Topic 2, cell division, meiosis. Meiosis is a type of cell division designed to make haploid cells called gametes. Gametes include sperm from testes and egg from ovaries. Meiosis occurs in 2 stages to produce a total of 4 haploid cells. Why are haploid cells necessary? Because then a sperm, which is haploid, and an egg, which is haploid, when they fertilize, they produce a zygote, which is 2N. So the ploidy is maintained in the somatic cells. During meiosis, a few things happen that are different from mitosis, starting in prophase I with synapsis. That's when the homologous chromosomes snap together. That's how we remember what it means. Synapsis, snap. This is when the homologous chromosomes pair up, they join at the centromere. While this is occurring, crossing over may occur. This is when they exchange genetic material or recombine alleles. The homologous pair forms a tetrad. This is only present in prophase I and metaphase I. That's what we call it when the 2 homologous chromosomes join together. The homologous chromosomes then separate during Anaphase I, and then the chromatids separate in Anaphase II. Mom says I can't do meiosis until I finish my interphase. Interphase is the stage prior to meiosis I. It's similar to mitosis. The germ cells will go through interphase. During this phase, chromosomes are replicated through DNA synthesis. The cell prepares for meiosis. So we have a nucleus, nucleolus, the DNA is in form of chromatin, and we have our 2 pairs of centrioles. Meiosis I is also known as reduction division, since chromosome number is reduced from 2N to 1N. So we can see in this picture, we have 2 chromosomes at the beginning of meiosis I, and at the end of meiosis I, we have 1 chromosome in each cell, which is maintained through meiosis II. Synapsis in prophase I is when the homologous duplicated chromosomes join at the centromere to form a tetrad. Tetra means 4. There are 4 sister chromatids joined together. In prophase I, the nuclear membrane disintegrates and the spindle fibers form. The chromatin coils up into chromosomes. 2 homologous or similar chromosomes come together to form a tetrad. This process is called synapsis. A tetrad consists of 2 replicated homologous chromosomes, or 4 chromatids. During this time, chromosomes are so close together, crossing over can occur. So both synapsis and crossing over result in entirely new genetic combinations not seen in the parents. So during prophase I, we have the centrioles that have moved to the poles of the cell, joined there by the asters. The nuclear membrane disintegrates. The chromosomes pair together to form tetrads. Spindle fibers form. And the ploidy of this cell is 2N equals 4. Crossing over means that the 2 homologous chromosomes exchange some genetic material so that the new chromosomes have some of the old and some of the recombined portion of their chromatid. In metaphase I, the tetrads line up at the equator on the metaphase plate on the same spindle fiber. Recall in metaphase of mitosis, all chromosomes lined up at the middle. So here we have the spindle fibers, and the pairs of chromosomes are lining up on the imaginary line we label the metaphase plate. The ploidy of this cell is 2N equals 4. Recall that in mitosis, the chromosomes line up individually because we want each side of the chromosome to move to the daughter cell, producing a daughter cell that is identical in DNA to the mother cell. In meiosis, the purpose of it is to produce haploid cells. So first of all, we have to pair the homologous chromosomes and then pull them apart so we get a haploid cell. In Anaphase I, the chromosomes pull apart. It's the homologous chromosomes that separate opposite poles. It's known as segregation, and it changes the cell from diploid to haploid, 2N to 1N. Individual chromatids in the replicated chromosomes do not separate yet. Okay, so remember, anaphase A4 apart. The reason that they are pulling apart is because the centrioles pull in on the spindle fibers, shortening them. Because they're attached to the centromere, it pulls the two sides apart. Telophase II, the chromosomes arrive at the opposite poles. Cytokinesis starts in animal cells. So we see the nuclear membrane reappearing. The big clue that it is telophase is that there is a cleavage furrow. We're drawing early telophase where we can still see the chromosomes in the cell. The entire cell, because it hasn't divided yet, is still 2n equals 4. In cytokinesis, 2 haploid cells form. Chromosomes may become chromatin and enter an interphase 2 in some cells. Haploid cells now go into meiosis II. So we have the centriole, nucleus, nucleolus. DNA is in the form of chromatin. This may occur, or the telophase may continue straight into prophase II. Prophase II, new spindle fibers form, the nuclear membrane dissolves, chromatin becomes visible chromosomes. So here's the spindle fibers, nuclear membrane disappearing, and we have just 2 chromosomes in each cell because the cells are now haploid, n equals 2. Metaphase II, chromosomes line up at the metaphase plate. Chromosomes here are made of 2 chromatids. So there we go, chromosome 1 and 2 on the metaphase plate, and same in the other cell. Anaphase II, the centromeres split, so we have segregation of chromatids or sister chromatids, which we now call chromosomes. So they're separating because the spindle fibers are shortening, pulling them apart. Remember that they are now referred to as a chromosome, no longer a chromatid, because they are no longer attached by the centromere to their sister chromatid. Each cell is still haploid. No, you never said there was a second anaphase. She's my sister. Remember, there's no cleavage furrow in anaphase. Okay, telophase II, chromosomes arrive at the poles. Cytoplasm starts to pinch in, forming a cleavage furrow. Cytokinesis starts. So we have the nuclear membranes reforming. Again, this is early prophase. We can still see the chromosomes. And the big clue that it's telophase is that there is a cleavage furrow. Okay, these cells are about to become n equals 2. Right now when they divide, here we go. And result is 4 haploid cells, each with a nucleus, nucleolus, and a haploid complement of chromosomes. And that's meiosis.